Take it away, Sergey.
Okay. Are you ready, Youngmin?
Yes, I'm ready.
All right, wonderful. It is my pleasure to introduce our today's speaker, Youngmin Park, who is a candidate for a position under the umbrella of artificial intelligence and computational medicine. Youngmin got his PhD from University of Pittsburgh under Bart Ehrmantraut, and he had several postdocs at UPenn, at Brandeis, and currently he comes from University of Manitoba. And today he'll speak about coarse-grained stochastic model of myosin-driven vesicles into dendritic spines. Please.
Great. Thank you very much for the introduction. And of course, thank you to the hiring committee for inviting me for this interview and giving me a chance to state my case. So yeah, as Sergey stated, this is the topic I'll be speaking about. And before I talk about this, I'd like to briefly go over my research just so everyone knows where I'm coming from. So apologies to the people that have seen this already, but yeah, they've seen these already. But the biological theme of my work is it's technically within neuroscience. That's if I had to give a word, that would be it. And within neuroscience, I look at how oscillatory neurons interact with each other and how they function, and how these interactions give rise to emergent phenomena. So things like central pattern generators that are responsible for rhythmic patterns for walking, breathing, swimming, those types of movements. Oscillators, coupled oscillators play an important role there. And there are many questions related to how central pattern generators are controlled. And that's one example of coupled oscillators. And I've thought about this question since I really started, and this is how I got my career started thinking about these types of problems. The other direction is very computational and has a data science flair to it. And this paper, Park and Geffen, this is a paper with an experimental person. And so this is kind of strong proof that I am someone that can walk and speak to experimentalists and successfully collaborate with them. And so this was a very computational model of a particular region in the brain, namely the auditory cortex. And I reproduced this model. I created this model that's capable of reproducing various results from various labs, specifically within optogenetics. And from that experience, I also gained some data science experiments analyzing gigabytes of neural data. And I've also applied other methods like hierarchical clustering to try to get some more out of some of that data. The third direction, which is more recent, has to do with molecular motor function. And I'm happy to say, I forgot to tell the search committee this, but this paper, Park et al. 2021, this has been accepted for publication in SciAm Journal of Applied Math, and Fay and Park 2022, this has been accepted for publication in the Journal of Mathematical Biology. And I'm continuing to ask questions about motor function, molecular motor function, while here at Manitoba. And the mathematical theme of my work is dynamical systems. I'm trained in applied dynamical systems, and I often use methods from bifurcation theory. I use perturbation methods because I'm often considering separations of timescales. And when it comes to stochastic models, I'm often computing mean first passage times. And this is by no means a comprehensive list. These are just things that I use most frequently and are kind of the most broadly accessible to a mathematical audience. And then the goal of this type of work is to coarse grain or reduce the dimension of a model, of a biological model, so that I can understand it to greater detail, generate predictions, do this kind of analysis that using the methods that are available to me. Okay. And the topics I'll be focusing on today are the molecular motor function, very closely related to this paper in SciAm Applied Math. I'll show some mean first passage time calculations, and I do some coarse grading to compute some quantities. All right, so without further ado, let's turn to the talk. I like to give a very concrete introductory background on where my research is physically. It always helps me personally when speakers do this, and so hopefully it helps you too. The story here starts in the brain, and if we take this, here's an example brain, it's a human brain, and if we take a slice, we get a picture like this, and this is one hemisphere of that slice. And the region I'm interested in general is this outer layer, which it's the neocortex, but I call it the cortex for short. And this is an interesting region because it seems to be responsible for intelligence. And there are plenty of animals that have heavier brains than we do. There are animals that have bigger brains in volume than we do. But really, no animal that we know of has as many neurons in the cortex as humans. And so there's this interesting correlation between cortical neurons and the size of the cortex and intelligence. So there's something interesting happening there. And how does it work? Well, we have to zoom in, and if we look a little closer, we have a picture like this. And this is a cell body stain, and it's just marking the cell bodies of all the neurons in this small region. We can see the scale is quite small. Here's the scale bar. And it's only a 2D slice. So in this brain is, of course, a 3-dimensional, and you can imagine just how densely packed this region is with neurons. Now, if we zoom in on one of these neurons, they're most likely going to be pyramidal. And pyramidal neurons are excitatory, and they make up most of the neurons in the cortex. So here's a zoom in on one of those. And in addition to the cell body, we can see the dendrites protruding off of the neuron. And so we can think of these as kind of the primary computational unit of the brain. Now, it might be a little hard to see in this picture, but these dendrites, there are a lot of them. And each of them they look a bit bumpy or a bit fuzzy. And this isn't an artifact of a low resolution image. These are actual structures. Here's an idealized image of a neuron with these structures they're with exaggerated structures. So the dendrites will receive incoming signals from other neurons, and those excitatory signals will often terminate on these protrusions, which are called dendritic spines. Okay, so we have dendrites, and the dendritic spines come off of the dendritic trunks. Each excitatory neuron receives on the order of 10,000 excitatory inputs. Most of them terminate on spines. So most of the brain is built of excitatory neurons, which receive most of its many inputs onto dendritic spines. And so it's not surprising that defects in these spines are implicated in important disorders like autism spectrum disorder, Fragile X syndrome, and Alzheimer's. And understanding how spines work and how they're maintained is an important question. Let's zoom in a little further, and I won't spend too much time on this slide just for the sake of time, but I want to just mention that their dendrites are often drawn as mushroom-like. That's the stereotypical image. They have this thin neck that opens up into a bulbous head. And people have thought about why this might be, that maybe it minimizes receptor loss, maybe there's a robustness in responses to incoming electrical signals. I won't talk about those, but because the specific problem I'm interested in is this question of vesicle trafficking. Now, what happens is that these surface receptors that receive the neurotransmitters, they diffuse off the spine head, they're sequestered by some process, and then they're transported back through this neck and hopefully to the other side. And this involves the trafficking of a vesicle. So this blob, it's some just a bunch of a lipid. It's a lipid bilayer that's kind of enclosed, and it takes some proteins and molecular motors. There's some active process that pushes it through this constriction, and eventually maybe it'll deposit the receptors on the other side. And people have thought about what this means, and in this particular study, they looked at whether or not how the constriction size of the neck affects the ability of a vesicle to actually pass through the constriction, and they used a constant force here. And they're only able to show that the vesicle either gets stuck or it passes through, depending on the radius of the constriction. And the problem here is that this is really far from the whole story, because if we look at experimental data, this is data from the University of Utrecht. We're looking at live cell light microscopy, so this is from living tissue. Note the scale bar, it's quite a small spatial scale. The black outline is a dendritic trunk, and these two bulbs are dendritic spines, and we're going to focus on the spine here. And apologies if you're having trouble seeing my mouse. For now, I can use a little laser pointer. And there's something interesting. There's a blob that comes in from the right. That is a vesicle. and it starts to enter the spine, but then it somehow escapes. And so there's this bidirectional movement. It's also incredibly difficult to get data like this. It's hard to just be at the right time and place to notice that there is a vesicle that does this. So in this field, data is actually very sparse. And this is where classical modeling techniques can be very, very helpful. But the point is here that movement is not necessarily unidirectional. Naively, I would have thought that you would want those surface proteins to go all the way through to the other side each and every time to make sure that the dendritic spine is properly excited by incoming neurotransmitters. But clearly that's not always the case. And the broad underlying question for all of this is how do molecular motors, which are the active process guiding that vesicle movement, and the geometry of the spine together affect vesicle entry? And in this talk, I'm considering subsets of this question, but this is the ultimate big picture question. Yeah. Okay. Right. Oops. Okay. So how do we tackle this question from a mathematical standpoint, and how do we understand things? Well, the first step, the first thing I do is to idealize the dendritic spine as a cylinder. And this might feel like an extreme assumption, but it turns out that this really isn't. What really matters is that there is a constriction. It doesn't matter if it's close to the vesicle, eventually will have an effect, but the point is that there has to be a constriction, and this constriction exerts a drag force. And I won't go into this too much in this talk. I'm always happy to explain, but this drag force it's a non-trivial drag force. And this is something that distinguishes my work from existing work on molecular motors. So just as an aside, a lot of existing studies on molecular motors, they neglect this drag force because they assume that the vesicle being transported by a motor is moving in some fluid, in some free space. In that case, the molecular motors are much stronger than the drag force, and you can neglect it. But here we're specifically looking at a constriction, and a constriction with a closed end. So the closed end of the spine is on the left here. The open end is on the right, so the vesicle enters from the right, and it'll get pushed by molecular motors, and it'll do something non-trivial, which I'll get to. But you can take my word for it for now that there is this non-trivial force that acts on the vesicle. And then the molecular motors, they're dominated by myosin motors in dendritic spines. So there are, and for simplicity, I assume that there are a species of upwards preferred myosin motors. That is, they prefer to push the vesicle towards the spine head. And there's a identical species that's symmetric, but it prefers to push the vesicle down away from the spine head. Okay. And yeah, right. So so please let me know if you have any questions about the setup here. Okay. For the myosin motors, this is what happens. This is an example of what happens with an individual myosin motor. So this was actually developed by Huxley back in 1957. That is the myosin motor model. And the idea is that we have a molecular motor that's being shown here. Its base is attached to the spine wall, and then its head is kind of it's looking to attach to the vesicle, and it attaches with some rate per unit time that's proportional to a parameter alpha. And then if the vesicle is moving in its preferred direction, which in this case is downwards, so this is a down motor, the molecular motor will push the vesicle or get pulled with the vesicle and then detach with some rate per probability per unit time that's proportional to beta. So it's a straightforward memoryless process. When the vesicle is moving in the non-preferred direction of a motor, then it'll still attach with some probability per unit time alpha, but it'll get dragged and then detach. It'll get stretched too much and then detach beyond some predefined position, some local position, capital B. Okay, so the dynamics here are less trivial. And the point of showing this is that the local motor positions for each of the motors that are available to attach to the motor or to the vesicle, they depend on the velocity. Okay, so yeah, we have all these details, but local motor positions, they depend on velocity. And right, that's something that we'll have to address in a bit. Mathematically, the way that I chose to describe this is to write down a variable that denotes whether or not a given motor is attached or detached. So if it's attached, it just takes the value 1. If it's detached, it's 0. And I always assume that there are a constant number of available motors available to attach to the vesicle, which I denote n sub x, for x being for down motors. And yeah, so little I will correspond to the down motors, little capital X corresponds also to the down motors. And then for the up motors, I use the same notation, but with J in place of I. And as I show these, it'll become more concrete over time. Apologies for overloading the notation already. But yeah, J and Y correspond to the up motors, upward preferred motors, and then I and X correspond to the downwards preferred motors. And from here, we can write down the model equations. dz dt, so Z is the vesicle position, V is the vesicle velocity, and here from prior work, we've justified that we can just use the center of mass for the position. So really Z is just a single point, pretty simple dynamics there. And then the second equation is we can think of it as a force balance equation. It sums the forces up from the down motors. Okay, so the down motors each have a position, xk, which depends on the velocity. We put it through a transformation to transform the local motor position to a force. For simplicity, I just assume a linear spring where k is a spring constant, so nothing especially complicated there. And also, there's nothing special about this choice for position to force. In the original work in Hoppenstadt and Peskin, they use a exponential function, but they also mention explicitly that the choice isn't anything special. Okay, but we can transform the local motor position to a force using this linear function f. And of course, I sub k, that's if a motor is attached, then that force will get summed up to the total down motor forces. Similarly, for the up motors, we have the same thing here. And then zeta, this is the confinement factor due to the spine geometry. So minus zeta v, that's a force that's acting against the velocity due to the drag, which is due to the confinement. And right, of course, I don't expect anyone to just remember all of this. And in fact, what I do is I take these equations, put them in Python, simulate them, and see what happens. And there's a little more thought put into it than that, but this is what I get. So on the left is a plot of the vesicle of the velocity as a function of time. And the gray bars denote a quasi-stable steady state. And this is from a mean field approximation from earlier work. And we see that the vesicle velocity fluctuates about that quasi-stable, the quasi-steady state. And then occasionally it'll switch to the other quasi-steady state before it switches back, and so on. And if we integrate the velocity, we can get an idea of how the position of the vesicle is changing. And we see that when the velocity is positive, that the position increases, and when the velocity is negative, the position decreases, and so on. So interpreting this plot, it's pretty straightforward. There are two important features that I want to capture when simplifying this model. And it is an expensive model to simulate. Because we're keeping track of every single local motor position for all the 100, 200 motors that are in the model. Okay, so there are two important features to capture when simplifying it. One is the existence of a bistable velocity. Interestingly, this comes up quite a bit in the study of molecular motors. You can have identical motor species that might attach in in a different way, such that the preferred direction is opposite of other attached motors. And things don't just lock with zero velocity, but you end up seeing a tug of war where one velocity dominates for a while and switches to the other velocity and back and forth, just like in this plot. And that's a classic result that people have seen time and time again since then. And the other and most important feature is the mean time to switch velocity. If I want to ask questions about translocation, the successful transport of the vesicle all the way through the cylinder to the other side, I better be able to capture the mean time to switch the velocity of the vesicle. Otherwise, otherwise I can't get an accurate idea of the probability to translocate and the mean first passage time to actually reach the other side, conditioned on translocating. So the goal of this work and what I would like to show you is how I've simplified this model. Like I mentioned, the agent-based model is expensive to simulate. You do need a relatively small time step, and there are a lot of variables to keep track of. So to understand how the molecular motor parameters affect things like the velocity switching times and questions about translocation, I better simplify this somehow. And this is the point where I'm now diverting away from the published or the accepted for publication SciAm applied math paper. And this is a different mass equation formulation that I've recently derived that I'm really excited about. So starting with the down motors, which again I denote with the capital X. If what I want to do is come up with dynamics for the total number of attached down motors. So I just take the sum of the I sub k's, and the sum is in the set. It goes from 0 to the total number of available motors. Okay, and we know that it grows with a particular rate, in a rate that's proportional to the number of unattached motors. Okay, and then the decay rate is a bit complicated. It depends on the velocity. Because of that if you're pulled in the non-preferred direction, you have that detachment. And so the faster you get pulled, the more quickly you detach. And that's why there's this velocity term in the denominator. But I'll simplify this in a moment. The point of showing this is it's just a little messy. And that's for the down motors and for all the down motors, and for the opposite species, the notation is virtually the same, but with J in place of I and Y in place of X. And it also has an equally complicated detachment term. So the velocity here, if we recall from a little earlier, maybe I'll go back for just a second. The velocity depends on this really complicated equation. At each time step, I would have to figure out what this equation, I'd have to solve this equation numerically to get the velocity. And that kind of defeats the purpose of what I'm doing, which is to find a simplified equation. So what do I do about the velocity? And what I've done recently in independent work is to rewrite the velocity as a function of motor states. And I'm skipping some details here, but it is under some assumptions, it's possible to approximate the velocity in this way. So I'll write the parameters in a moment, but this approximation does become more accurate as the total number of available motors increases. It's not so I don't know how quickly the accuracy converges, but I have something. It's just to say that I've thought about this more than just writing it down. The Tx and Ty, these are the mean attachment times for each motor. And I'm going to assume that they're roughly 1 over beta, 1 over the detachment rate beta. And this is granted, it's a very strong assumption, but I do this because it significantly simplifies the model. And it ends up giving actually a pretty good estimate in the end, when I put everything back into the original equations, the simplified equations. Okay. And if a more precise velocity estimate is desired for whatever reason, that is the subject of the SciAm applied math paper. The reason I'm not considering it here is that it's a PDE. There's a PDE involved in the calculation, which must be computed numerically, so it's less analytically tractable. And what I wanted to do was write down something that was analytically tractable. Okay. Capital A, that's just the local motor attachment position. Zeta was the confinement factor from before. K is the spring constant. So yeah, we just have the velocity in terms of both the motor states, but also in terms of the molecular motor parameters, the microscopic parameters. And the point of this slide is to say that I can write velocity as a function of motor states. And if there are any errors that come out of this in the end, well, I've made my assumptions very clear, and we can always come back and adjust that if needed. Okay, so there's the velocity equation again, and the detachment rates for the down motors, we had this complicated expression, and it actually becomes simpler, and this is the simpler form. Now delta just depends on the motor state's ij as opposed to the velocity, and this ends up being just very straightforward to compute. Okay, and likewise for up motors, we have a very straightforward expression, and I'll be writing delta yij for the detachment rate of up motors, delta xij for the detachment rate of down motors. And right, okay, so just to be concrete, before v had to be computed numerically, but now what I've done is written an approximation for v where I don't have to compute it numerically, and that simplifies calculating these rates. Now I have everything I need for a master equation. Okay, so given state ij, there's some rate that determines how quickly I leave the state ij, and that's a sum of the various attachment and detachment rates. And then there are various ways I can come into the compartment ij that's based on the various attachment and detachment rates of the neighboring compartments. And if we let the vector p be the vector, or have these entries, so if you were familiar with the discrete Poisson problem, the numbering I'm using here is just the same as that. So I'm taking the columns of the matrix Pij and just stacking them on top of each other to create this vector P. Okay, so it's just a pretty straightforward stacking. If I do that, I can rewrite this master equation as a linear system. dp dt equals q transpose P, where Q is a generator matrix that's a function of all these on and off rates, attachment, detachment rates. Okay, and explicitly, Q, I can write this down explicitly. Q is block tridiagonal, and the various matrices, I can write them also down explicitly. So there's this explicit equation I can get for the master equation in a very straightforward way. Okay, and I tend to go a little too quickly sometimes, so I'm just trying to pace myself here. Okay, yeah, and each of these matrices in the block tridiagonal matrix Q transpose, they each have very nice forms. The MJs are each tridiagonal, the DJs are diagonal, and then the lower off diagonal terms, these are just a constant times the identity matrix. Okay. So it makes calculations really straightforward. And there's something to note here, pretty standard results, but the matrix Q corresponds to a strongly connected graph. It's possible to go from any state, any total number of attached motors I and j, to any other total number of attached motors, which implies that Q is irreducible. And this implies that there exists a unique steady state P star that satisfies the system of equations. And it's possible to solve this analytically. I think it would be very helpful to do that. I will explain that later. But for now, just to get something out, I will solve this numerically. Okay, so this is just a representative set of parameters. This is from a textbook, and it was actually for muscle myosin, which is a little different from, so it's from skeletal myosin, which is different from transport myosin. But in the end, these actually give reasonable values when it comes to vesicle velocity and translocation times. So I'm not too worried about these choices. The confinement factor here was just chosen, and this corresponds to some constriction that's not too tight, not too loose, somewhere in the middle. And I'm using 100 motors for each species, so 200 motors total. If I simulate the agent-based model and plot the total number of attached motors and normalize that distribution, I get a bimodal distribution, which makes sense, right? We saw that the velocity was bistable, and that means that underlying that the molecular motors are also, there's going to be a distribution that's bimodal. And if I look at the master equation that I just derived and compute the steady state distribution, I get a really nice match between the two. And so the peaks are in very similar places, if not the same. And the magnitudes are very similar. And in fact, for the agent-based model here, I used a pretty dang small sample size. This is one realization for a relatively short time. So I anticipate that the match would become much better with more simulations or with more simulation time. Okay, so the steady state distributions match. Well, what I really want to get at is the mean first passage time. And for this part, I'm using pretty standard off-the-shelf methods, but I've had a lot of fun with this. So what I want to do is, if I have some Markov chain with, and I want to compute the time to switch from state I to state J, I do the following calculations. One, I take the same generator matrix from before, and remove column J and row J. And this corresponds to J being an absorbing state. I can compute the survival probabilities by writing first the solution Pj. It's just the solution Pj is the matrix exponential TQJ. And then if I multiply the right side with the vector of ones, I can get the survival probability at time t for each node that's not J. Okay. Then to get the distribution of the hitting times, I take the derivative multiplied by -1. So it's a distribution as a function of time. And if I define the expected hitting times as this vector, where T1J is the expected hitting time from node, the first state to J, Tnx ny, that's the expected hitting time from that, where all motors are attached to state J, then I can do a calculation to compute what this looks like. First, I take the average of the distribution of hitting times. So that's where the t comes from, and that's where the integral comes from. It's just an expected value. And pretty much everyone in the room, I think, is looking at this and just screaming integration by parts. And that's what we do next, integration by parts. And we arrive at this term. And from here, we can use, the QJ ends up being invertible. And so we can use standard methods for integrating matrix exponentials, and we arrive at this term. So what's really nice is that we don't need to compute any integrals. There's really no numerical anything that we have to do. We just have to compute the inverse of Q sub J. And Q, again, that's the generator matrix with row and column J taken out. Once we do that, we multiply the vector by the vector of ones, and we can get the hitting times. Okay. So same parameters from before, so same representative parameters from before. Here's the formula that I'm using. And I use this to calculate the time to transition from state 010 to time 0. And I got something on the order of 0.31 seconds. And it's pretty dang close to the agent-based model, which is on the order, if you recall, it's on the order of 0.22 seconds. Now, if we're looking at these two numbers in isolation, it seems like a relatively, there's a relative large error between them. But my goal was actually I didn't state this, but my goal was to get within the same order of magnitude as the agent-based model, because I tried some other approximations, which I also didn't show, namely the Langevin equation, where the estimated time to switch the velocity was it was 4 orders of magnitude different from the agent-based model. So the fact that, right, so that I guess is a low bar, but still, I'm pretty happy with what I've gotten here. The other thing is that with this approach, what I've written gives us a lot of potential for analytical solutions. And I did this stuff numerically, but a lot of this, I think it is possible to be able to write down explicitly what the expected hitting times are for an arbitrary absorbing state J. And so we can, and same thing for the steady state, I think it can be done analytically. And this is a potential project for any grad student that might want to join, because what's the contribution? Well, once we do that, we can actually get an instant sense of how the microscopic parameters affect the emergent behavior. And I think that's an incredibly useful thing and can lead to good biological questions and predictions. So still work in progress. I'm going to keep going through with it because I'm really excited about this. But yeah, if anyone's interested in this kind of thing, then I'd be happy to I'd be happy to work with them. And okay, and then if for some reason all these reasons are not enough, and you feel like the time to switch estimate here is not close enough to the agent-based model, then I actually have a much better estimate, a very, very good estimate in my SciM applied math paper. And again, I didn't consider that here because it's more numerical and less analytically tractable, but yeah, but it's there. Okay. So now I'm going to go back to the contents of the original paper, the paper that I was originally going to present on, because I think this next part's really cool. And this part is good if you don't necessarily care about the microscopic parameters, if you don't care about the details of what the motors are doing and how the rates affect the emergent dynamics. We can do simplification. What I noticed is that in the original agent-based model, if I plot the distribution of waiting times to switch velocity, I get a histogram that's shown in blue. Okay, so it's underneath these curves. And as a kind of a first order test to see what distributions best describe this histogram, I took all the distributions available in Python on the order of 80 something distributions. I fit them each to the histogram and also computed the Kolmogorov Smirnov the Kolmogorov-Smirnov, divergence test. And there, the smaller the number, the better the fit between two functions that are tested, in just intuitively speaking. And I found that the generalized exponential and the exponential function do a fantastic job relative to all the other functions that are available. And just for simplicity, I said, let me just kind of run with the idea that, look, this is approximately exponential. Technically, it looks like it's a generalized exponential distribution. But let me just try running with just assume that it's an exponential distribution. Then we can recast this entire problem into something much simpler, namely the telegraph process. And what we have is a vesicle that moves with some roughly constant velocity. For this part, we'll just assume that the vesicle has a constant velocity, and then it occasionally switches to the opposite velocity. And the waiting time is roughly exponentially distributed. And just by definition, this is a telegraph process. And a lot of work has been done to understand telegraph processes. So a lot of tools are available to us. Here's a schematic of the problem. We have a one-dimensional domain. Z equals 0 is an absorbing end. Z equals L is also an absorbing end, but this corresponds to the top end of the spine. The left corresponds to the open part of the spine. And the vesicle will move around in this domain and keep switching velocities until it either successfully hits the other side or fails to hit the other side because it hits the left boundary. Okay. For some reason, I mean, people had looked at mean first passage times of a very similar setup, but they always considered the mean first passage time to just exit this interval. They never considered, of the 13 or so references I found, they didn't consider a conditional mean first passage time or a conditional probability where they say you never hit this side, we'll condition on not hitting this side, then what's the probability of hitting that side? So that was a novel contribution that we made in the actual paper. Okay, but yeah, now the problem is much simpler, and we can ask straightforward questions. So, for example, let's let E plus of Z naught t be the probability to escape the right side of the interval up to time t, where Z naught is some initial position and plus indicates an initial positive velocity. And we'll consider two time intervals, one on a very short time interval and then the other on a possibly much larger time interval. Delta t, of course, is small. And if we look at the first interval, at the end of that, we have a new position. The vesicle I've moved with a positive velocity, and so I'm at a new position that depends on the vesicle velocity, the delta t, and the initial position. I call that Z nu. And at that point, the orientation either switches or it stays the same. That's just kind of trivially true. So and then it'll switch from positive orientation to negative with probability lambda delta t, where lambda is the switching rate of the exponential distribution. And then +1, it's that the probability you continue with that positive velocity, and that's the opposite probability. So 1 minus lambda delta t. Okay, and I'll call that sigma nu. And so if I look at E plus, so the probability to escape the right interval given initial positive velocity starting at z naught at time t plus delta t, this is the average over the orientations of E plus at z nu. And writing the average explicitly, we have that probability to maintain the orientation. Z nu is I've just written out the definition of z nu, plus the probability to switch orientation times the probability of reaching the right side given initial negative velocity at that given the initial position z nu. This step is probably the most non-trivial. So again, if there are any questions about it, please feel free to ask. And the next steps are more straightforward. What I do next is Taylor expand in delta t and collect the lowest order terms and collect the delta t terms. And I end up with an equation like this. And if you're a grad student, there might be a pattern emerging here. There's a small delta t, there's a t plus delta t. You don't have to say it if you don't want to, but anyways, yeah. So I could just give you the answer here. I rearrange this equation. I subtract e plus of z naught t on both sides, and then divide by delta t. And if you've taught Calc 1, taken Calc 1, this is starting to look very familiar. And from this point, I take the limit as delta t goes to 0, and I end up with a PDE for E plus. Now, E plus is to get the actual quantity that I want, the probability to escape the right interval over any time. I take the limit as t goes to infinity and end up with a ODE. I do the same process to derive an ODE for E minus using the exact same method that's written here. And in the paper, I call them boundary conditions. They're really more like initial conditions. But this is saying E plus, that's if you start at the right boundary with positive initial velocity, you're guaranteed to escape. You just instantly leave. So that probability is 1. And if you start at the left boundary with initial negative velocity, you'll instantly leave there and you'll never reach the right side. So this E minus of 0 is 0. And together, these three equations, it's possible to solve this in a pretty straightforward way. And I'll get to that in a moment. But here are the equations. We can do a very similar set of calculations to find the mean first passage time for this telegraph process. And if we define S as E times T, we can get these equations. And if there are already grad students that are interested in this kind of work, I highly encourage you to try and derive these equations using the methods that I use for the probability. And it uses basically the same logic. So it's really good practice. And we can justify the boundary conditions based on the probability and the mean first passage time T plus at L. And this quantity is from, if you're at the right boundary with initial positive velocity, you exit immediately. And so the time to exit is 0. That's what that's saying. Okay. And I'll conclude with this figure. I'll say a few, just a small number of things after this, but this is the main thing. So what I did is compute the mean first passage time to translocation over different lengths, and as well as the probability to escape to successfully reach the other side. And what I found is that the length, as the length increases, the probability decreases. And in fact, the probability gets quite small. And I found this surprising because I thought that even for longer dendritic spines, the cell would want the probability of translocation to be high. but this is saying otherwise. And the mean first passage time, correspondingly, it increases as a function of length, which that makes more intuitive sense. The gray squares here, I will ignore them, but these are some numerical simulations. And here I had to use a cluster to run these. But this is the other master equation that I considered in the paper, the original master equation that I didn't present on. And that was just to confirm that the simulations match the telegraph process. Okay, and there are all kinds of questions that come out of this. So this already is a bit of a prediction. This is saying that the longer your dendritic spine, the less likely you are to translocate. And if I kind of take that as a truth, just pretend that that's true for a moment, then that seems to imply that, or rather, it makes me wonder if cells are somehow controlling, if a cell wants to be less excitable, is it somehow making its dendritic spines longer so that it receives less surface proteins and therefore becomes less responsive to inputs? I don't know. But this is that's a hypothesis that kind of follows from these graphs. And likewise, if a cell wants to become more excitable, maybe it makes its dendritic spines shrink more. And is this really what's happening in cells? Yeah, I don't know, but that is a prediction, and maybe one day I'll be able to find experimentalists that with the right tools to start looking at questions like this. Okay, and then just a brief note that in the model, the quasi-steady state velocity for the representative parameters, it was on the order of 120 nanometers per second. Translocation times varied from 50 to 70 seconds for spines on the length of 500 to 800 nanometers. And in the literature, the velocity of myosin motors are on the order of 200 to 450. So I'm within the same order of magnitude. I'm okay with this. And translocation times, it's on the order of 50 to 75 seconds for spine lengths that are 750 to 1200. So again, within the translocation times that the model produces is pretty similar to what's in the literature. Okay, so at least things are consistent. All right, so to summarize, I derived a master equation that relates the microscopic motor parameters to the switching time in vesicle velocity. And to do this, I had to rewrite the velocity in terms of the attached motors. This allowed me to compute the steady state distribution and the mean time to switch velocity, which, right, and then from there, one can further simplify the problem as a telegraph process. From there, it's possible to compute the probabilities and mean times to translocation. And for future directions, I really want to see what the analytically computed steady state distributions and the mean time to switch velocity looks like using the master equation that I introduced in this talk. I really want to understand also how the microscopic motor parameters affect those quantities, as well as and also how it affects the question of translocation. That's something that's not possible with the paper as it's published. But yeah, this new work that I've shown, I think it makes it possible. So thanks to Thomas, my excellent postdoc advisor at Brandeis, Prashant Singh. He's a co-author in this paper. I've never met the guy. I have no idea what he looks like outside of pictures. We've only communicated by email, but I guess that's the beauty of doing applied math. We can kind of work and build collaborations wherever we want. I was paid by a T32 while at Brandeis, and thank you all for your attention, and I'll take questions.
All right, thank you for this very exciting talk. Please, if anybody have any questions.
Okay, I have a question. Hey, Youngmin. Well, it's nice talk. But in the beginning, I'm not very sure that I get the major question you want to address. Or I'm not in the field of computational neuroscience. So you want to use the model to, for example, calculate the quasi-steady state velocity and the translocation time, or?
Yeah, the short-term goal was to compute so the quasi-steady state velocity, that was actually done already. And I have a good way of doing that because I have a mean field version of the model, which of course I didn't present on, but that was published in 2020. So there is a way to do that already. But the mini question that I'm looking at for this talk, the sub question is exactly that. It's the question of how do you compute the translocation time, and also, to be fair to you, I didn't quite do that with the main model, the main master equation that I introduced. That work is so new that I haven't had a chance to apply it to that question. But in the end, what I went back to was the telegraph, rewriting everything as a telegraph process, and that at the time I was writing the paper allowed me to answer that question. So yes, that's exactly the quantity that I wanted to compute. Yeah.
Okay. But in the end, you also show some data in the literature. Is that from the experiments or?
Yeah. So this is the two citations that I have. Where is it? Yeah. So this is data from experimental literature. Yeah. And this is, I had to eyeball this. So this paper is very nice. It shows time-lapse images of vesicles that are actually going through dendritic spines. And what I did was measure, oh, this vesicle seems to have moved this many nanometers and between these two frames, which are, however many minutes apart. And that also gives you a sense of how sparse the data is in this field. That was the best that I could work with. But yeah, it's from the experimental literature.
Okay, thank you. Well, I have another couple of technical questions, but I can wait.
Okay.
Can I ask a hypothetical question? So in your model, you basically consider a single vesicle traveling through this neck, right? What if you had multiple vesicles in the same, competing for the same set of up and down motors? What would, your model can easily be generalized, right? To actually consider this as a possibility. So if you have two vesicles entering the same neck approximately at close times, and then what they get stuck, they block it completely, or one actually helps to push the other one through?
That's a really good question. And
or is it even relevant? Because in I don't know how many vesicles there are to, on the time scale to be near the same neck, or what do you call it, spine?
Yeah, the neck of the spine. It ends up, so sure. I think it, so my understanding is that this event is relatively rare. It's hard to observe this. But still, I think that's an interesting question to entertain. And that question is I suspect it's actually very complicated, because what I'm actually doing now is looking at a similar question where we have nodes that are forced by molecular motors that are connected by linear springs. And this is a similar, in some sense, this is similar to what you're asking, because if you have vesicles, they're going to interact somehow. And so that there's that non-trivial interaction. And yeah, even looking at the simple case of having a linear spring between two nodes, there's a lot that's unknown, and I'm actually having a lot of trouble. Well, not trouble, but yeah, it's been very interesting mathematically, and there's a lot of work, and so I can't necessarily say concretely. Well, actually, I will say this: they found in prior work with the nodes and springs that velocity tends to they tend to move together. So there's a cooperation. So there might be some non-trivial transients, but then after a point, there's one velocity that's chosen, and they just move together. So it's possible that would happen in this case.
Thank you.
Thank you.
Any other questions, please?
I think, oh, sorry. You want to go?
No, go ahead. Go ahead.
Okay, I was just going to say, so kind of going along, you were just mentioning about like the springs, and I'm just thinking about the different assumptions that you have made for the model. And one thing, it sounds like data is somewhat limited. And so I guess, could you give a little bit of intuition or just feedback in terms of when you are putting together the model pieces, how much of it is kind of mathematically convenient, how much of it is experimentally approximating, and then what's your vision for the future? For like, are you planning on trying to make these different model pieces more accurate according to the data, if you have it, and like kind of what your plan is for that?
Right. Yeah, that's an excellent question. So let's see. Yeah, okay. So the main mathematical convenience is actually assuming that the vesicle is purely spherical. That's the strongest simplifying assumption. It's not even assuming that the dendritic spine is a cylinder. So I was explaining this to Philip earlier, but I'd love to explain this again, because this assumption is actually, it underlies everything that I've been doing. So if an object that's close in diameter to this constriction is entering a closed space that's filled with a fluid, the hypothesis, the current hypothesis, which we don't know if it's true or not, we might never know in our lifetime, is that as this vesicle enters, there's a backflow of fluid that escapes out the sides of this vesicle. Okay, does that make sense? So if we have like a glass of water and a tennis ball that's close to the diameter of the glass, and we push the tennis ball in, there's going to be water that spurts out the sides. And this allows one to simplify the Navier-Stokes equations, and it ends up being and that's where this confinement factor and this drag comes from. It's because that fluid is coming out the sides, and there's more details to it, but that's the fundamental assumption. So you can imagine that backflow of fluid doesn't really depend on what the shape of the vesicle or the geometry of the spine, as long as there's a constriction that's going to happen. And everything I'm doing really rests on that assumption, and I don't know if that's true. So that's the key thing that we would have to verify somehow eventually. But there is an earlier study that my postdoc advisor did, more computational, but he did some estimates of the various the viscosity and he made some estimates on what the fluid is like inside a cell, and he got numbers that are him and I actually, we consistently get numbers that are consistent. They don't contradict anything yet. So yeah, I just don't know. And then as for making things more complicated, for now I'm going to continue using this spherical vesicle, because again, the assumption is that fluid backflow idea, and yeah, that's either true or not, and for now I'm just going to assume it is true.
Thank you.
Sure, thank you.
I see Luca had a question. Luca, go ahead.
Oh, sorry, my 14 months old raised the hands. It was an accident.
Oh,
Sorry. Sorry, but very nice talk. Sorry for that.
any other questions? Okay, let's thank Youngmin again. Thank you.
Thank you.
And so in 15 minutes, right, we meet for the exit interview.